Okay, hello GCSE biology students, and welcome back for this next lesson in the Inheritance and Genetics series. This is going to be quite a dense one, packed with lots of examples, because the truth of the matter is we can't predict which example, right, is going to be pulled up onto your GCSE. Right, it could be one of the ones specifically mentioned on the syllabus, or it could be a different one that is given to you just out of the blue. But the principles are all the same. So we're going to have a look at family trees and how we interpret those on exam papers, inherited diseases, and screening. So cracking straight on then, that's the bit of syllabus. As it says here, look, what a lot of students should be able to, telling you what you should be able to do, right? They are using probability, using genetic crosses, using direct proportions, simple ratios, lots of math skills, and make predictions using theories around probability. Now, if you don't know how to use a genetic cross yet, make sure you've watched the previous video, which will explain all that, because I am going to assume a knowledge of that lesson in this lesson. Okay, so we looked at a cross, a simple Punnett square genetic cross. Now, I think that's the best way to do these kind of genetic crosses. It's the clearest way, right? The idea of the little crosses with the two sets of gametes, and if that sperm meets that egg, and so on and so forth. You might see this kind of layout as well, right? If you draw like this in an exam, perfectly fine, still worth all the marks. Mark scheme is still the same. Parental genotype, work that out, that's a mark. You work out your gametes, you fill in the possible combinations for the offspring, and then you work out their phenotypes and answer the question. Okay, that's fine. But don't be thrown if you get something like that in a textbook or on a worksheet or in an exam. Okay, if we want to do this as a Punnett square, however, right, we can convert quite easily between the two. So we just take the parental genotypes here and pop them on either side of the square, and we end up with something like that. Okay, so you can see that the two are the same. So that might be that parent, that's that parent. There are our gametes, so that's the gamete layer. The gametes are down here. And then here's our genotypes. Now, we don't know what the question is for this example, but the last point, as I said again, would be to answer the question. Okay, moving on then. So if you get family trees, right, this is possibly the hardest way you could be asked a genetics question. Okay, so here's a family tree, an initial family tree, and this one's about tigers. Okay, so in this instance, this square here means male, and the circle means female. That's standard family tree nomenclature. Okay, so squares for male, circles for female, and then colours, shaded or not shaded, can mean whatever the key says that it means. So in this instance, the orange ones are our orange tigers, and the white ones are our white tigers. Okay, so obviously this is a family tree of tigers that has got some white tiger genes in it. Okay, so the kind of questions you might be looking to answer go as follows. Which are the youngest tigers? Which is the mother of the youngest tigers? What's the relationship between the tigers on the bottom row and the three on the middle row? Which allele is dominant? Is orange dominant to white, or is white dominant to orange? And how might you represent the alleles when writing out the genotypes? What might you use? Okay, what is the genotype of tiger A? What is the genotype of tiger B? And work out what the genotype of all the other tigers could be, and draw a Punnett square. So these are a selection of questions you might get on this family tree. So what I suggest you do is pause the video now, work through those ones, and we'll get the answers shortly. Okay, we've done that. If not, pause. Okay then, so first one, the youngest tigers. The way these family trees work is they go oldest at the top to youngest at the bottom. It basically comes down the page like that. That's the sort of flow of genetics going that way. Which is the mother of the youngest tigers? So these are the youngest tigers here. We follow this back, okay, and we're trying to find a female. So the mother, the circle there, is A. Okay, so the mother of the youngest tigers is A. Okay, what's the relationship between the tigers on the bottom row? So these ones, G through P, and these ones D through F. Well, they are siblings. They're brothers and sisters. Okay, if you see this kind of sort of comb-like affair, they are brothers and sisters. They're siblings. Okay, now you've got to use a little bit of intuition here. What allele is going to be dominant, orange or white? Well, you should have come to the conclusion that it's orange is dominant. Reason why, look, we've got an orange parent here, okay, so an orange colour parent with white offspring, okay? So this parent here presumably is homozygous recessive, and this one is heterozygous. So that one's always going to donate a smaller white gene, and this one is going to donate is going to have a dominant orange gene, orange allele showing up, and in the cases of G, I, K, and N, has donated a recessive that was hiding away in that individual A there. Okay, so genotype of tiger A, we reckon should be a dominant and a recessive, heterozygous. Okay, B, homozygous recessive. Right, as if that was homozygous dominant. Okay, if that was homozygous dominant, well then every single one of these would be white. Just wipe out the orange colour. Okay, so if we look at some of the answers for this one, right, I reckon those are our genotypes. Okay, and therefore anything with our white phenotype is going to have to be homozygous recessive, just like the blue-eyed example in the last one. And then if we work it through, these individuals here must have received their mother's dominant gene. Now again, we're using O's here, which is not for orange, which is not the best letter to use if you're handwriting it, because it looks the same in upper and lower case. So we're donating orange here, orange there, and so on and so forth. Okay. Now what we can see, we can count this up: 1, 2, 3, 4 individuals who are white. 1, 2, 3, 4, 5, 6. Now that obviously is not exactly 50-50, but the probability of each one being either white or orange would be half, okay? Because the dad here is definitely going to donate a little O, mum either a big O or a little O, okay? And that's the example I gave you in the last one about my eye colour between blue and brown, okay? So if we were doing a genetic cross look, here it is. So if we had 2 of the orange ones crossed like that, we would have that standard, right, 1/4 to 3/4 orange to white Punnett square that we did in the last lesson. Okay, so here's an example directly off an exam paper with 2 of the questions I've picked out underneath. So this is what I mean about giving any random genetic disorder at all. Cram, C-R-A-M, is the example. It's an inherited condition which causes muscle breakdown. It doesn't matter one bit if you know what Cram is or not. Completely irrelevant, right? In fact, you just need to know the breakdown product enter the urine, making it dark red colour. That's how you know. Okay, doesn't matter. So here's the table here. Look, you can see each numbered person. The reason that they're numbered is you would be expected in your answer when it says give evidence to use those numbers to say, well, individual this and this have this child, and this child can't possibly be that. Okay, so give evidence from the diagram that Cram is caused by a recessive allele. Okay, well, if we have a look, the best bit of evidence that I can see here that couldn't be explained any other way is this. Look, 7 and 8, a man and a woman who don't have Cram, C-R-A-M, they themselves do not have Cram, have a child that does. Okay, so the only way that could work is if they were both heterozygous, if they both had one dominant and one recessive. These guys either got 1 or 2 dominant alleles, or got lucky. This one here was the 1 in 4 probability chance of getting 2 recessive alleles. So this is a boy with Cram. Okay, so you would say in that answer, 7 and 8 have managed to produce 10 who has got Cram when they don't. Okay, and that suggests, that's evidence that it's caused by a recessive allele. Lots of people in this question go up here and have a look at 1 and 2 producing 5, 6 and 7. Now that's kind of evidence. The idea that 1 might be homozygous dominant, in which case it doesn't matter that this person's recessive because they're all going to get a dominant one. But we could explain that the other way around. If we just looked at this, okay, then Cram could be dominant, and this could be recessive, and these could just be lucky. So this could be a heterozygous person, right? But they could have 1 recessive the other way around and give that to these 3 all at the same time. So 50% chances coming up at the same time. So it's not impossible the other way around with 1 and 2. The one that is impossible is 7 and 8. So the best, clearest evidence is 7 and 8 produced number 10. Okay? Right, next one. None of person 2's children have Cram, explain why. So that's using this person here. Well, knowing this is recessive, this person here, so the dad has donated a dominant allele to each individual in 5, 6 and 7, meaning in their phenotype they don't show Cram. But as we now know, this person must be homozygous recessive. They must be like little c, okay? These people will all be heterozygous. They'll all be carriers. They'll all have one working gene, one working allele of that gene from their dad, and one not working allele of that gene from their mum. Okay? So we can work out their genotype as well from looking at their parents. Okay? That's a kind of high-level exam question. Right, and before we finish the family tree section, so do pause the video, go back, have a look at that, make sure all those answers make sense, and just for interest's sake, right, to have a look at the family tree I've put up here from the ancient Egyptian pharaohs. Okay, now when you start to look at this, you'll see some quite unusual things. Right, now the ancient Egyptian pharaohs believed that they were descended directly from gods. They were gods among people, and therefore to outbreed from the family was diluting royal and divine blood. So you can see what they did here and how many new people actually entered the gene pool, right? Actually entered the family tree. So this is not so much a family tree really as a family stump, I would say, or family trunk. Okay, not recommended, because of course any genetic problems they will have build up and are more likely to be concentrated in this family. Okay, so do pause and have a little look, and it's fascinating to go and read about the genetics of individuals like Tutankhamun, all that kind of stuff, and the genetic issues that the pharaohs suffered because of this attitude towards breeding. Okay, so that's the first section done on family trees. Let's talk diseases then. Okay, let's talk genetic diseases you have to know about. So, many diseases come from genes. There are many inherited diseases, okay? Most are recessive, which means they can hide away in generations, but there are some that are dominant. Now, there are some examples you have to know. Cystic fibrosis and polydactyly are the examples that are on your spec. Huntingtons and sickle cell anemia are just other good examples. As you've seen with the Cram example there, they could throw anything at you and expect you to apply your knowledge to these examples. So for every single one of these, we're going to go through, we're going to answer the question, is it a dominant or recessive gene that causes this, dominant recessive allele of the gene that causes this? We're going to have a look at the Punnett square to show how it's inherited. We're going to look at the symptoms, the current treatment, and any potential future treatments. I'm going to go quickly bang through these 4 examples. In the full knowledge, you could get any other examples when it actually comes to your exam. Okay, so we'll look at cystic fibrosis first to start with. Now, cystic fibrosis is caused by a recessive faulty gene. Okay, so I've done the classic, 2 parents here who have one dominant working gene, allele of that gene, and one allele of that gene that is the cystic fibrosis allele, so it's recessive, so it's the small one. It affects this gene called the CFTR gene and protein, and that's involved in transporting chlorine ions, specifically salt ions, in the lung and in the gut. So if we look at the inheritance, it's exactly the same as the inheritance of blue eye colour that we did last time. Dad makes his sperm, half of them are going to be dominant, half of them are going to be recessive. Mum makes her egg, half of them dominant, half of them are recessive. You do the table, if that sperm meets that egg, that person is a homozygous dominant, so that person doesn't have any cystic fibrosis genes, so they can neither have it nor pass it on. These people here, look, that sperm and that egg, and that sperm and that egg, have a dominant gene there like their parents, which will mean that they don't have this disease themselves because they've got one allele that will make the protein properly, so they'll be able to transport chlorine properly, but they are what we call carriers. They can pass this gene on in the future, okay? And then the 1 in 4 unfortunate person, double C down there, little c, that's the person with cystic fibrosis, that 1 in 4 chance. Okay, so hopefully that's familiar to you because it's just like the blue-eyed and brown-eyed thing with the cystic fibrosis gene, recessive gene, taking the place of the blue-eyed gene, okay, blue-eyed allele, right? So we actually know quite a lot about cystic fibrosis because it's a very well-studied disease. It's actually quite common, certainly in Western European populations. Right, it's quite common and arguably the most common inherited disorder in the UK. Okay, so we've got cystic fibrosis sufferers down there. Now we know quite a lot about it, as I say. We know where it is in the genome, so that's chromosome 7, and we know this gene exists on Q branch 31 to 32. As you should know from the last lesson, that's the gene locus. That's the place on the chromosome where the gene is. And as with every other gene, we've got 2 different alleles, which is where we get our tables from. Okay, these 2 different alleles. So we're going to go a little bit extension now. Now you don't have to necessarily know exactly what that particular gene does, apart from it messes with the transport of chlorine, messes with your lungs and your gut. But I'm going to tell you exactly how that works by the medium of amazing PowerPoint animation. Now I know with my PowerPoint animations you will believe you're inside a cell. You're not. You're healthy. It's fine. Don't worry about it. Okay. So this is a cell membrane here, and this is a working functional CFTR protein coded for by a working functional CFTR gene, and it lets chlorine move in and out. Now this brings in diffusion, which you should also know about from earlier in your GCSE. So the chlorine, look at them go, they can move straight through the membrane, no problem, until there's the same amount of chlorine on either side of the membrane, and we've reached what we call equilibrium. Again, a term you should be familiar with. Now, if the gene for that protein is broken, right, then it changes the shape of the protein. If you go back 2 lessons and remember the thing about DNA changes the amino acid, changes the structure of the protein, okay, which means that our chlorine now can't fit through the hole. So what we end up with is we actually end up with loads of chlorine on one side of the membrane and hardly any on the other. Now that is like putting salt on a slug or putting salt on anything else or sugar on fruit salad, and it causes osmosis. Again, you should remember that from earlier in your GCSE, and it actually draws water across the membrane into there. Now if this is the lungs, this is the tubes of the lungs, the trachea, the bronchi, all that kind of stuff. Again, another part of the GCSE you should know. And this is the inside of the cells. And we're basically sucking water out of the lining of your lung tubes, okay? And that causes this thick, sticky, dehydrated mucus in the tubes of the lungs that makes for a really, really difficult job breathing. Also works for the gut as well, which makes digestion difficult. Now, so symptoms, what's the effect? Thick, sticky mucus, which clogs the lungs and the digestive system. You can see a scan of the lungs over there with all the mucus in it. Causes breathing trouble, of course. There's no cure at the moment, but it's treated by physiotherapy and similar drugs to asthma to try and loosen the lungs and get that mucus moving, even though it is thick and sticky, okay? There is gene therapy on the horizon in the future for this, trying to get that working gene into the cells, right? But that's obviously, that may well be the first genetic therapy that's available on the NHS, but it's a little way off yet. It's close, but we're not quite there yet. Okay, right. Next example. So this is your next example. Now you should be able to spot what's wrong with this picture. Right. This is a condition called polydactyly. Right. Poly meaning many, dactyl meaning digits. Think pterodactyl and all that kind of stuff. Right. Dactyl is digits, dexterous, that kind of root. So polydactyl. You've got an extra digit on your finger. Now this one is a dominant allele of a gene, okay? So if you get 1 copy of this, you get an extra digit, right? So in this example, Dad has got an extra digit or had got an extra digit. Mum does not. So Mum's homozygous recessive. Dad's heterozygous. Now you can work out the probability here, look, right? That person there gets 1 big P, and because this is dominant, that means they get an extra digit. That person there gets the dad's sperm with the recessive one in, which is normal, which means they don't. 5 fingers, okay? That one there gets an extra digit, that one there doesn't. So because it's dad with this sperm and egg thing, right, with this 2 sperm thing, dominant in one, recessive in the other, what's the probability of getting an extra finger in this scenario? Well, 2 out of 4, or a half. And this is exactly the same as the inheritance of gender, the inheritance of biological sex, rather. Okay, we talked about in the last lesson. Okay, so it's the dominant P there gives you an extra finger. Okay, now of course the treatment for that is pretty obvious, right? It's not a serious disease, not life-threatening like cystic fibrosis is. Most people will just have a cosmetic procedure, cosmetic surgery to just remove the finger. Okay. Now, obviously, you might think, well, why not? Why have 6 fingers? It's great. Having 6 fingers doesn't necessarily mean you've got the wiring, if you like, the nervous bit to actually control all your fingers separately. Right? Usually, it's one finger that's split and bifurcated. Okay. So usually, it's very simple. Early in life, there's a surgical removal of the extra finger, and no harm, no bother. Okay, so that's a difference between cystic fibrosis. Cystic fibrosis is recessive. You need 2 copies to get it, and it can be fatal, and there's no cure. Okay, polydactyl, dominant, you only need 1 copy. It's not fatal, but most people just have it chopped off anyway. Okay, so those are the 2 that you actually have to know, and you can probably see why they've been picked as 2 contrasting examples. So I'm going to give you another couple of examples that fall down this same route. Huntington's disease, you may have heard of. It's another relatively common inherited disorder, and it's a terrible inherited disorder, really, really nasty. Okay, this one's a dominant one, so just like polydactyl, this time I've replaced H's for P's. If you get 1 copy of this, it kills you, right? It's a neurodegenerative disease, and it kills you, okay? So there are no carriers of this disease, okay? You can't have it hidden away. It's a dominant gene, right? So Dad here, look, has got his Huntington's here, right? And in the same way as polydactyl, it would be a 50-50 as to whether the children got it. Now, you might have spotted this question straight away the minute I said if you get 1 copy, you die from it. Because then the disease this destructive, 1 dominant allele of this gene, and it kills you, how does it not die out in the population? How does Dad get to pass this gene on in the first place? Okay, well, the answer to that one is that it doesn't actually show up till middle age. And this is where this disease is particularly terrible, okay? So the people who develop the symptoms of Huntington's, right, already have children, right? So it might show up in their 40s, 50s, 60s, whatever it might be. So we've then got the scenario, and I have met and spoken with and taught some students who've been put in this position, and it's a terrible position to be in, right? If your dad or your mum develops Huntington's, then you know from that simple table it's a 50-50 chance whether you got that gene and whether if you did, it will kill you, okay, at some point in your life. And that's a terrible situation to be in, okay? If your mum and dad develop it, well, you can work through the probabilities there. So if they've both got a dominant gene by bad luck and come together, then obviously you've got a 75% to 100% chance that you'll get it, okay? Which is very, very bad, okay? And the more generations it goes on in a family, we're not going to go into why, this sort of repetition of gene sequences, the more generations it goes on from a family, the worse it gets, and the earlier and earlier in your life it comes on. So terrible, terrible disorder, right? Much more serious than polydactyl. It's a dominant disorder, but it's that middle-age thing that means it persists in the population. And can I commend to you the work of various charities like the Huntington Disease Association, who support families and research for people with this condition. Okay, so to give you an idea of what it is, this is a section through a brain of someone who died from Huntington's, and as you can see, there are lots of gaps and holes in it. So the cell basically causes brain cells to degenerate. So symptoms of that would be anything associated with dementia, right? So clumsiness, jaw clenching, loss of coordination, slurred speech, swallowing, uncontrolled muscular contractions, walking difficulty, and so on and so forth. Okay. Now, I have heard tell that this degeneration of the brain can be so serious that in a post-mortem of someone who died from this, the post-mortem worker thought that the brain had already been removed because so much of the middle of it had already disappeared. Okay, terrible disease. Obviously, if you hear about this and know about this, do consider helping those charities out. Okay, same goes for sickle cell anemia. It's another really common one in the UK. Okay, it's common in certain groups within the UK. This one is recessive. Okay, it basically means your red blood cells don't form correctly. It's a mutation in the hemoglobin gene, and you can see from the pictures there why it's called sickle cell anemia, because half your blood cells basically end up collapsing on themselves and are called sickle shaped. Okay, so they form in these sickles rather than the lovely biconcave discs you know about from earlier in the GCSE, and that stops oxygen being carried. Right? Obviously, then, if you can imagine those little sickle-shaped cells trying to push their way through your little tiny capillaries in your fingers and your toes, that can be intensely painful. So it's the standard cross. There we go. You've got no sickle cell, not a carrier. Carriers here, or what we call so-called sickle trait, because in the case of sickle cell anemia, if you've got one, half your blood cells are sickle-shaped and half are not. So this person is perfectly unaffected. These two have got half the blood cells, and this person here will have all of their blood cells sickle-shaped and will not be able to survive without blood transfusions to get working red blood cells in there. Okay? So, now the sickle cell is a bit weird, which is why I include it, because it's a very strange one. There's a sickle cell mystery, and this links to the next part of your GCSE syllabus. So by the Punnett square, about 50% of the population maximum should be carriers of sickle cell anemia, even if it's quite common in the population. Being a carrier obviously gives you symptoms, as I just said, so half your blood is that sickle shape and half is not. So logically you'd think that that's a bad thing, so really those people should be less likely to reproduce. So you'd think the frequency of the gene would go down across the generations. In reality, in some areas it's more like 70 to 80% of the population carry the gene. Well, why might that be? Okay, and this is where we get into an evolution point. Okay, now the solution to this is that it actually gives an advantage elsewhere. So yes, it's a disadvantage in carrying oxygen around your body, which makes you less able to run, carry, have aerobic fitness. But malaria, again, as you know from another part of your GCSEs, we're bringing in lots of different bits of the GCSE here, is a disease of the blood. It's a parasite, protozoa that gets into the blood and parasitizes the red blood cells. If half your red blood cells are sickle-shaped, your body is going to respond to that. It's going to try and destroy the sickle-shaped ones and manufacture new blood cells, right? But what all it's doing is it's constantly doing that from the wrong instructions, because one in two of those alleles is for sickle shape. So all it's doing is making more sickles and destroying them. Okay, but that just means that the blood has a really high turnover, so our malaria parasite can't get a foothold as easily in someone with sickle trait. So if you look at the maps, and I think this is a fascinating example of human evolution, if you look at the maps here, we've got areas where malaria exists in green, and we've got areas where the frequency, a heat map here of the frequency of the sickle cell hemoglobin gene. So as you can see, look, down here, the heart of that sort of malarial region is where you find the highest frequency of these sickle genes. So it's an advantage to be a carrier or have sickle trait. One sickle gene protects you from malaria, so the big B people are likely to die of malaria because it's very common in those areas. The little b people will die of the sickle cell anemia without blood transfusion. So actually the people who survive the best are the heterozygotes, right? The people with so-called sickle trait. Even though it makes them a little bit sicker, it protects them from something that's arguably as big a threat. So this is a fantastic example of modern evolution in humans, and it's why this topic links perfectly into the next one, which is about evolution, speciation, and genetic change over time. Okay, so that's the diseases section done. I'm going to leave you this to ponder, and we'll discuss it more next time. Screening. So we now actually have the ability, because we know about all these genes, to borrow a cell from any person or embryo and screen it, look for these alleles of these genes. So we can now look at the genes of either prospective parents or of an embryo itself, that's the picture there is showing, and screen it for these genes and say, right, you two, if you have a baby, you've got X percentage chance of having sickle cell anemia, Huntingtons chorea, cystic fibrosis, this, that, and the other. Loads of different genes are available now. Okay, and you can do that at the parent level, and then they can take a choice as to what they want to do. Then you get into questions of, if you do that at the embryo stage, well, what's the option here? If you tell a person, you know, your child's going to have this horrendous disease, if you let that embryo grow into a child, then that child will have a horrendous disease. The option is termination, or the option is using IVF to create and screen embryos. And you can see we enter a whole world of ethical conversations to be had at this point. So evaluative questions on the GCSE could ask questions like, is it right, or should we screen or evaluate the use of screening for cystic fibrosis versus polydactyly? This is why those two very contrasting examples have been picked. Okay, now what you think is entirely up to you, but I can tell you from a GCSE point of view, you need to stick to the science, and you need to give, if you're asked an evaluate question, a balanced view, and if you're asked for a conclusion, you draw a conclusion. If you're not, you don't. So these are fascinating and interesting and important conversations for the age that we live in. I'm not going to go into them now. Obviously, if we were having a conversation in class, that would be lovely. We could have a debate, but there's plenty of material out there for you to go and research and listen and come to your own opinion and your own conclusion on that. Okay, so that's it for today. Bit of a long one. I know lots of material in there, tons of dense stuff, but it's all good. It's all good material. So I'll see you next time as we get towards the end of the inheritance topic. Thanks very much.